So in this video, we are going to talk about parasitic helminths. And so just a reminder, our helminths are our worms. And because worms are animals, worms are heterotrophic. Remember that heterotrophic refers to other feeders. These are organisms that have to consume other things in order to survive. They have to take in carbon as organic compounds. So they have to take it in as their macromolecules. When we talk about worms, adult worms can be what's referred to as dioecious. Di means two, and that means that the male reproductive organs are in one individual and the female reproductive organs are in another individual. So there are two worms that have to come together in order to reproduce. Other worms are what we call monoecious. Mono refers to one. These are worms that are hermaphroditic. One animal has both male and female reproductive organs within the same organism. When we talk about our helminths, helminths fall into two main phylums. And so Platyhelminthes, think of flat. These are flatworms. We have our cestodes, which are tapeworms. They are flat and they appear segmented. We have our trematodes. These are our flukes. These are flat, unsegmented worms. And then we have our nematodes, phylum Nematoda. These are our roundworms. And so we're going to talk about different examples of different types of parasitic worms. So what you're going to want to do during this is you're going to want to fill out the table that goes along with this lecture. And so there are several components to the table. You'll notice that the organism is already given to you. Then we have our disease, our classification of our worms. So are they a cestode, a trematode, or a nematode? Is the worm monoecious or dioecious? What are the stages to identify on the exam? We are going to talk about the infectious stage and the mode of transmission. We're going to talk about the approximate size of the worm. And the size of the worm, when we talk about its size, it's not so much that I need you to memorize exact numbers, but to get an idea of scale. Are these very small worms or are these much larger worms? And so that's why I'm going to go through and give you the approximate size. We will talk about the symptoms. So what are the symptoms of the disease that they cause? What is the treatment? How do we treat this? And then the last column is going to be our diagnosis and any other additional notes. Now, again, a good rule of thumb is if I put it on the slide, it's probably something that you want to make sure is down on your table. Some of the things will pop up as animations. Others might already be written on the slide and I might just underline them. And so if it's on the slide, it's probably a good thing to know. And so just keep that in mind when you're going through and you're studying this information. And so let's go ahead and get started. The first worm that we're going to talk about is Anaerobias vermicularis. Anaerobias vermicularis causes the disease anaerobiasis. Another name for this is pinworm. And so if we talk about pinworm, it's the most common worm infection in the US. It's not the most common in the world, but it's the most common worm infection in the US. And so that's something good to know for this, this worm. In terms of its classification, Anaerobias is a nematode, which remember means that it's a round worm. You'll notice if you look down here, these are the adults. Notice there are two adults. One is male and one is female. So this worm is going to be dioecious. It has separate male and female worms. In terms of stages to know on the practicum, what you want to know is you want to know the adult worm. And again, to give you an idea about what you're looking for the adult worm is about the size of a grain of rice. So they're fairly small worms. They're not particularly large. And then the egg, what you're looking for on the egg is you'll notice that one side of the egg, so overall it has kind of an oval shaped, but one of the sides of that oval is flattened. You'll notice that this side up here is more rounded, but this bottom part here is more flat. So that's what you're looking for when you're trying to identify the enterobius egg. You're looking for that one flat side. And so those are the two stages that you are responsible for on the exam. So the egg and the adult. So now we're going to move on to the infectious stage and the mode of transmittance. And so the egg is the infectious stage. This is the stage that causes the disease. And the way that you get pinworm infection is through the swallowing or inhalation of the eggs through contaminated clothing, bedding, or fingers from scratching, for example. Poor sanitary habits of children make them especially likely to transmit the disease. And so let me explain a little bit about how this disease works. And so the adult worms are going to develop in the intestines. So when you swallow the eggs, right? You swallow the eggs and then they're going to hatch and develop in the intestines. Now, male and female will be in the intestines. They will reproduce. And so the way that the female is going to lay her eggs is, remember that they reside in the intestines, and the female at night is going to crawl down and out of the intestine, and she is going to lay eggs on the perianal region. And so the perianal region is the region between your anus and your private parts. So that's your perianal region. So at night, she's going to come out, she's going to lay her eggs on the perianal region, and then she goes back up and into the intestine. Now, one of the symptoms of the disease, and we'll talk about the symptoms in a minute, but one of the main symptoms is going to be an itchy rectum. So especially for kids who are infected with this pinworm infection, right? They wake up in the morning, they have an itchy rectum, right? Their butt itches. And so kids being kids, they don't think anything of it and they scratch. Well, when they scratch, they end up with the eggs on their fingers, in their bedding, in their pajamas, and again on the fingers. And so if they're scratching and then they go around and they touch things in the house, well, those eggs can survive outside the body for up to 2 weeks. So if they touch, fomites, so counters, etc., anything that they touch could potentially be infected with these eggs. And if you touch that surface and then you eat, let's say, well, now you're going to swallow the eggs. So the mode of transmission is through swallowing or inhaling these eggs. And so again, you get it through contaminated clothing, bedding, or fingers. And so this is how you end up with the infection. Now, the adult, again, are found in the intestines. And when the female lays eggs, you don't have to memorize this number, but just to give you an idea, when she lays her eggs at night, she lays between 4,600 and 16,000 eggs a night. So she is very fertile. She lays a lot of eggs, which could give rise to potentially a lot more pinworm infections. In terms of size of the adult, again, the adult worms are about the size of a grain of rice. Male and female look different. The female is going to be the larger of the two, and she is approximately 1 centimeter in size. The male, on the other hand, is going to be smaller. He is approximately 0.5 centimeters. So he is smaller than the two in terms of male and female. But the identifying feature for the male is he has this reproductive organ that's called the spicule. And so that is his reproductive organ. And so it looks like a little curly Q, whereas you'll notice that the tail of the female is very straight. The male has its spicule. It has its reproductive organ. And so that is how you identify male versus female. So now let's talk about our symptoms. So for symptoms, many people with pinworm do not have symptoms. They don't need treatment to cure the infection. The infection may go away on its own, although this may take up to 14 weeks, which is the equivalent of 14 weeks is the equivalent of 2 pinworm life cycles. The common symptom, if somebody does, again, is going to be the itchy rectum. The eggs are going to irritate the rectum, and it's going to cause the symptom of the itchy rectum. In terms of treatment would be albendazole, trade name Albenza, or mebendazole, Vermox, and these are typically taken 2 doses 2 weeks apart. And that's because the pinworm medicine does not kill the eggs. It only kills the larval stage. And so if any eggs were laid during that time that it took the drug to have an effect, those eggs could hatch and then lead to an infection again. And so what somebody has to do is they have to take the doses, 2 doses 2 weeks apart. So the second dose is to kill any larval stages that might have hatched during that initial treatment. There are over-the-counter medicines that you can actually take. Parental, it's otherwise known, there's a couple versions of this, but it's just called pinworm medicine or Pin-X. There are several variations of this that are over-the-counter. And so that is also a treatment. Same thing, you would want to take 2 doses 2 weeks apart to try and make sure that you got rid of even those eggs. Now, in terms of diagnosis, so remember that the female is going to lay her eggs on the perianal region. And so for diagnosis is going to do something called a cellophane tape test. And so what they're going to do is you're going to take a piece of this cellophane tape, and let's say that a doctor wanted to test if the patient had pinworm. So they would send the patient home with a piece of this cellophane tape. And the instructions would be first thing in the morning, so before going to the bathroom, before changing out of pajamas, first thing in the morning, take that cellophane tape and touch it to the perianal region. And then give that tape to the physician or the lab and have them look for the presence of eggs under the microscope. If they see those eggs, then the patient has a pinworm infection. And so this is how this is going to be detected. It might take multiple tests before they actually see the eggs. So oftentimes they might not just test once, they might test repeatedly. But again, it's going to be this cellophane tape test looking for eggs on the perianal region. And so that's for Enterobius vermicularis. So this, again, most common worm infection in the US. This is the one we see the most often. So what we've provided for you is some images from the specimens that you would have seen in the lab so that you can see what our slides would look like. So this slide is using 40x total magnification. And This is our Anaerobias vermicularis female. So notice that she doesn't have a curlicue. Her tail is going to be pointed, whereas the male would have a little tiny curlicue. But this is not the male. This is the female. She has a pointed end. The other specimen that you need to know is the egg. Again, the egg is going to have one flat side. So this other edge is rounded, whereas this one edge is flat. And you can see that on both of these eggs that are viewed with 1000x total magnification. And so this is what the eggs will look like. You're looking for, they are kind of oval shaped, but they have one flat side. And so that's for Anaerobias. You need to know the egg, and you need to know the adult female. The next worm that we're going to talk about is Ascaris Lumbricoides. Ascaris Lumbricoides is going to be the most common helminth infection in the world. That's an important fact. So the most common helminth infection in the world. Over 1 billion people in the world have this worm. So about 1/7 of the world population is infected with Ascaris. This is a nematode. It's a roundworm. Notice if you look at the picture, the adults, there's a male and a female that are separate, which tells you that this worm is dioecious. There we go, dioecious. Stages that you need to know on the exam. You need to know the adults. And I'll show you in a minute, the adults are actually going to be found in a jar, in a bottle, because they're quite large. They're not the size of a grain of rice like Anaerobias. They're quite a bit larger. So you need to know the adults, and you need to know the eggs. And so what you're looking for on the egg is that the edge of the egg has what's referred to as mammillations. Think of it like scalloped edges. So the edge of the egg has this kind of scalloped appearance, and that scalloped edge is referred to as mammillations. So, in terms of diseases, I forgot to tell you, the disease that it causes is called ascariasis, as well as it causes something called Ascaris pneumonia, which we'll come back to in a little bit. So ascariasis and Ascaris pneumonia are the diseases. How do you get this infection? So the transmission is going to be ingestion of fertilized eggs in fecally contaminated, uncooked food or water. So how does this happen? Well, this can happen when hands or fingers that have contaminated dirt on them are put in the mouth. So if the dirt is contaminated with the eggs and then you put it in your mouth, that can happen. Or by consuming vegetables or fruits that have not been carefully cooked, washed, or peeled. And so this is also going to be common or found in association with poor personal hygiene, poor sanitation, so maybe not access to clean water, and in places where human feces is used as fertilizer. Because many times you need a fertilizer to provide nutrients that plants need to grow. And in developing and third world countries where they might not have access to fertilizer, commercially available fertilizer, they might use human feces as fertilizer. Now, for humans, we have the adult worms developing in the intestine. And so those worms can reproduce, and the eggs will come out in the feces. So if that person is infected by this worm and their feces is used as fertilizer, well, now those eggs are on the plant. And so if you eat that produce without it being washed or peeled or cooked, right, Then you're going to ingest these eggs and you're going to end up with this infection. In terms of size, the males, again, are going to be the smaller of the two. He is approximately 31 centimeters. So notice 30 times or 31, whatever, approximately times the size of Enterobius. The females are about 35 centimeters in size. Again, just like for Enterobius, what you're looking for to differentiate between male and female, you're looking for the presence of the spicule. And you will see this when I show the example of the worms that we use in the lab so that you can actually see what the spicule looks like. But again, it's like this little curlicue, and that is the worm's reproductive structure. In terms of the symptoms are usually asymptomatic. If symptoms do occur, they can be light, and they include abdominal discomfort. Heavy infections can lead to intestinal blockage. So this could lead to a severe case where there's so many worms in the intestine that it prevents food from moving through the intestines. And this can also lead to impaired growth in children. They get malabsorption. They're not absorbing their nutrients because these worms are occupying so much of their intestinal wall that it makes it difficult for them to absorb their food. And as a result, patients might have impaired growth. Other symptoms can include a cough due to the migration of the worms throughout the body. And we will come back to the cough in just a moment. In terms of treatment for Ascaris Lumbricoides, same drug as Enterobius, but typically for 1 to 3 days. So again, the Albenza, the Vermox, those would be the same diseases. What you're going to see is that for all of the nematodes, all of the roundworms that we talk about, those are the drugs that are used. So that same drug, that albendazole, the mebendazole, those drugs, those are used for all of the nematode infections that we are going to talk about. Now, in terms of diagnosis and additional notes, there are a couple things that we need to talk about in terms of the life cycle of Ascaris. And so what we want to understand is, again, it's going to be the ingestion of these fertilized eggs. So notice that when you ingest it, that worm is going to go down through the stomach to the intestine. The egg is going to hatch in the intestine and develop into the larvae, the juvenile stage. That larval stage is going to burrow out of the small intestines. It's actually going to make its way through the small intestine and into the peritoneal cavity, and it's going to penetrate the lungs. It's going to develop in the lungs for approximately 10 to 14 days. And so when the larval stage is in the lungs, that's what's going to cause the Ascaris pneumonia. And so coughing, again, is going to be one of the symptoms. And so there can be lung damage in response to this larval stage being in the lung. The patient might end up with a secondary bacterial infection, and the pneumonia can be fatal. And so if they get this Ascaris pneumonia, that could be very devastating for the patient. Now, when the larval stage is developing in the lungs for 10 to 14 days, those juveniles are then going to make their way up through the respiratory tree to the esophagus, and then they're swallowed and they go back down into the stomach, and then again into the intestine. And so they're going to then develop in the intestines as the adults. And so the eggs are going to be produced because the adults reside in the intestine. They're going to produce the eggs. A female can produce approximately 200,000 eggs per day. 200,000 eggs per day. She produces a lot of eggs. In terms of diagnosis is by identifying eggs in the stool sample. So again, looking at the stool sample for the presence of these eggs. Multiple stool samples have to be used sometimes because a patient might be infected, but they might not have the adult worms that are far enough along in their development to reproduce. Between 2 to 3 months are required from ingestion of the infective eggs to oviposition, so the ovulation by the adult female. And the adult worms can live about 1 to 2 years. So that's the lifespan of one of these worms, typically 1 to 2 years. And so again, because it takes 2 to 3 months from the time you ingest it until the time where the females are reproductive, that's why you have to often do multiple stool samples, because again, it might be that she's just not producing eggs yet, but you're still infected. And so this is why multiple stool samples might need to be used. But again, we are looking under the microscope for the presence of these eggs in the stool sample. And so this is Ascaris Lumbricoides. So these are the specimens. These are in glass bottles. This you can see is the male. Here is the spicule, the little curlicue at the end, whereas the female has straight pointed ends. Notice that the female is the larger of the two. So she is larger than the male. But again, the male has that spicule. It has that little curlicue. I'm going to erase this so you can see it better. He has that little spicule, which is his reproductive structure. So for this, you have to be able to differentiate between male and female. And so you're looking for the presence of the spicule. Here are 2 different images of Ascaris lumbricoides eggs. The one on the left is showing at 400x total magnification. The one on the right is showing it at 1000x magnification. And so again, what you're looking for on this one is you're looking for these mammillations. The scalloped edge is the characteristic for the Ascaris egg. That's how you're going to identify that stage. Next, we have Nicator Americanus. Now, I've heard this pronounced a variety of ways. Nicator Americanus, Necator Americanus, whatever one you want to say. I'll just say Nicator Americanus. And this causes what we call a hookworm infection. And an estimated 576 to 740 million people in the world are infected with hookworm. So almost 500 million or more than 500 million people are infected with this worm infection. This is going to be, again, a nematode. It's a roundworm. And again, notice you can see in the picture, the adult, there are 2 worms there, which tells you that this worm is dioecious, right? Male and female are in separate worms. In terms of stages to know on the exam, stages to know. So you need to know the adult. So this is the head of the adult, and its mouth has these little hooks which allow it to attach to the intestine. So you need to know the adult. You need to know the falciform larvae. This is the stage that is infectious, which we'll talk about in a minute. And then you need to know the egg. And what you're looking for on the egg is the jelly coat. So let me go ahead and put this. So you're looking for the jelly coat. It almost looks like this, this little jelly around the edge. And again, on the adult, you're looking for the hook. So those are the 3 stages that you are responsible for Nicator. Now let's talk about mode of transmission. So if a person is infected with a hookworm infection, the adults reside in the intestine. And so the male and the female are located in the intestine. And so the human will pass eggs in the feces, because when adults reproduce in the intestines, they're going to produce eggs, and the eggs are going to be deposited into the soil. Now, if an infected person defecates outside, so near bushes, in a garden or a field, or if the feces from an infected person is used as a fertilizer, then the eggs are deposited on that soil. Now, the eggs can then mature and hatch 1 to 2 days, and this releases a stage that's referred to as Adoptiform larvae. And so this is going to be the first stage after the eggs. So they're going to mature, they're going to form the Adoptiform larvae. And then after 5 to 10 days, those larvae mature into a stage that's referred to as the Falciform larvae. The Falciform larvae are the infectious stage. This is the one that causes disease in humans. So how do you become infected? Well, you become infected by walking barefoot on contaminated soil, and the Falciform larva penetrates the skin. So it's going to penetrate the skin and it's going to burrow into the skin. The infective larvae, those Falciform larvae, can survive in favorable environmental conditions for about 3 to 4 weeks. So they can survive in the soil for about 3 to 4 weeks. People living in areas with warm and moist climates and where sanitation and hygiene are poor are at risk for a hookworm infection if they walk barefoot or in other ways that allow their skin to have direct contact with contaminated soil. So in the US, this was seen more prevalently in the South, where people would go out barefoot in the woods and they would step on these larvae and end up with this hookworm infection. However, we don't see that as much in the US these days. In terms of size, the females, again, are going to be larger. She is about 1 centimeter. Males are between 0.5 to 0.9 centimeters. So what you want to realize is that this worm, the Nicator, is approximately the same size as Anaerobius. They are both approximately the size of a grain of rice. And so they have a very similar size. So now let's move on and talk about symptoms. So if we're looking at symptoms, itching and a localized rash are often the first signs of infections. And these symptoms occur when the larvae penetrate the skin. So at the site where the Falciform larvae burrows into the skin, the patient might end up with this localized rash. A person with a light infection might not have symptoms. They may be asymptomatic. A person with a heavy infection may experience abdominal pain, diarrhea, loss of appetite, weight loss, fatigue, and anemia. And the physical and cognitive growth of children can be affected because, again, we can get an instance where we might not get absorption of nutrients in a proper manner. Now, in terms of anemia, anemia is a condition that happens when you don't have enough red blood cells. And so what happens in anemia is that patients become very tired and fatigued because without enough red blood cells, they're not transporting oxygen effectively. If they don't transport oxygen effectively, because hemoglobin is in red blood cells, if they don't transport oxygen effectively, the cells in their body, their tissues, are also going to have a problem getting access to oxygen, and therefore they're going to have problems making ATP, because again, you need O2 to act as the final electron acceptor in order to make ATP. So if there's not enough oxygen getting to the tissues, we're also not producing enough ATP. And so patients experience fatigue, they're tired because they're not able to make enough energy to meet the demands. Now, why is that you become anemic? Well, because one of the things that happens is that the adult worms, and I'll talk about how it moves through the body in a minute, but the adult worms can attach to the lining of the intestine by their hook. They grab on and they take a blood meal from the patient. And so 50 worms inside the body can suck 1 cup of blood per day. And so that is what can lead to the anemia that we see. In terms of treatment, again, I said that the treatment for the roundworms is the same. So the same drugs as enterobius, the albenza, the Vermox, etc., for 1 to 3 days. And so those are some of the drugs that are used. Some of the drugs work by selectively inhibiting the synthesis of microtubules in parasitic worms, and that blocks the uptake of glucose and other nutrients. And so that's how these drugs can be effective. So moving on to our diagnosis and additional notes. So we need to kind of understand how the organism moves through the body. So again, on contact with a human host, the falciform larvae are going to penetrate the skin. And when they penetrate the skin, they're going to be carried through the blood vessels. So notice I'm telling you the route that it goes through the body. So the larvae penetrate the skin, they travel to the blood vessels, then to the heart, so you can see the dotted line to the heart, and then to the lungs. And so when they're in the lungs, they can penetrate into the pulmonary alveoli. They, again, like Ascaris, when they're in the lungs, they can go up the bronchial tree and to the esophagus where they are swallowed, and they go back down and into the intestine. And so they will develop into the adult in the lumen of the small intestine. And so that is where they're going to attach to the intestinal wall. And that, again, is what's going to cause that blood loss. Most adult worms are eliminated in about 1 to 2 years, but the longevity may reach several years. In terms of diagnosis, just based on everything we've learned so far, what do you imagine the diagnosis would be? And so, diagnosis, identifying the eggs in a stool sample. So again, taking a stool sample and looking for the presence of the eggs in that stool sample. The entire process from skin penetration to the adult development, so forming the adult in the intestines, takes about 5 to 9 weeks. So that's about how long it's going to take for them to develop into the adults. Females release about 9,000 to 10,000 eggs per day. So a lot, not as much as Ascaris, but still a very good amount of eggs. So what we're looking at here is we're looking at a hookworm adult. This is a different organism, but it's going to look very much the same as Nicator. And so anterior is referring to the head. So on the left, you're seeing the picture at 100x. And then we zoomed in, so it says 100x detail. And you can see these hooks, those little hooks on the head, which allow the worm to attach to the lining of the intestine. And so that's why it gets its name, hookworm. It has these hooks which allow it to hook on and attach to the intestine. This is the posterior or the tail. So again, it just has this pointed end. So our nematodes are round worms with pointed ends. This is the pointed end. Here is the falciform larva. So again, here is the head. This is the tail. Now, you can't be asked by looking at just this picture to determine if this is the falciform larva or if it is the anaerobious female. They're too difficult. They look very similar. So just be aware that there won't be any questions that make you try and distinguish between those two very similar organisms. So here we are looking at the hookworm egg. And so what you can see, the arrows pointing to that jelly coat, what looks like the jelly coat around the egg. And so that is going to be the distinguishing feature for the egg. Remember that the anaerobious has one flat side. Ascaris, the defining feature is the mammillations. And the hookworm egg, the defining feature is going to be that clear jelly coat. So next we're going to come to Schistosoma mancini. And Schistosoma mancini is a trematode, which is a flat, unsegmented worm referred to as a fluke. And schistosoma causes schistosomiasis. And more than 200 million people are infected worldwide. If you look at the adults, there are 2 worms, which tells you that the worm is going to be dioecious. It has a male and a female that are separate. There are several stages that you are responsible for the exam. One would be the adult. The adult, the male, is the wider of the 2. And you can kind of see there's like a little, what looks like a fold. And that's referred to as the gynecophoric groove. And that's like a little pocket where the female can reside and they can continually mate over and over again. And so the male has the gynecophoric groove. He's wider. He has this little pouch. And the female is going to be the thinner of the 2. So you need to know the difference between the male and the female. You need to know the egg stage. And the egg stage has what looks like a surfboard fin. So it has this little fin down here. That's the defining feature of schistosoma eggs. And you need to know the cercaria. The cercaria has a forked tail. It reminds me of like a mermaid's fin. So think of the egg and the cercaria. These are stages that are found in water. And so they have their little fins or their tails to help with motility. So we have our egg and we have our cercaria. So there's 4 stages. You need to know the adult male, the adult female, the egg, and the cercaria. Those are the 4 stages you need to know on the exam. Now, let's talk about how you become infected with schistosoma. So the way that schistosoma occurs is that you have to come in contact with contaminated fresh water that contains certain types of snails that carry the parasite. So how does this all happen? Well, let's say a person is in the water and this person is infected with schistosoma. The schistosoma is going to be released from the body, from the human body. The eggs are going to be released in either human feces or urine. So let's say somebody is out in the water and they urinate in the water. That is going to release eggs into the water. The eggs hatch into free-swimming larvae referred to as Mericidium. So we have these Mericidium, and the Mericidium is going to penetrate this type of freshwater snail. Now, this freshwater snail is the intermediate host. Remember back to when we learned about protozoans, we learned about definitive and intermediate hosts. Intermediate hosts are where the asexual part of the life cycle occurs, right? So this is asexual. So the water has to contain these snails. So this is only found in certain regions. So the Mericidium is going to penetrate the snail. The Mericidium is going to reproduce in the snail, and it's going to form Cercaria. Cercaria is a larval stage that is released from the snail. So the Cercaria are released from the snail. Again, here's the head. We have its forked tail. And these free-swimming Cercaria penetrate the human skin. And when they penetrate the human skin, they lose their tail. Only the head of the Cercaria is going to penetrate the skin. So the transmission is when the Cercaria penetrate the skin of persons who come in contact with contaminated water while wading, swimming, bathing, washing clothes, etc. So if you are in water that is infected, the Cercaria can penetrate the skin and can cause the infection. And so this is how you could get schistosoma. In terms of size, it says here, so the females are about 15 to 20 millimeters in size. That comes out to 1.5 to 2 centimeters. So about double the length of Enterobius, for example. The males are slightly smaller in terms of their length, but they are wider. In terms of the symptoms, most people have no symptoms when they're first infected. They are asymptomatic. However, within days after becoming infected, they may develop a rash or itchy skin. Within 1 to 2 months of infection, the symptoms may develop into what looks like a flu. So fever, chills, cough, muscle aches. And then the symptoms of chronic schistosomiasis, so the long-term effects, could include abdominal pain, enlarged liver, blood in the urine, blood in the stools, as well as problems passing urine if you get blockages. The treatment is going to be a drug called praziquantel. It's a prescription medicine that's taken for 1 to 2 days to treat infections caused by various schistosoma species, because there are multiple schistosoma species that cause variations of schistosomiasis. In terms of diagnosis and additional notes, let me put some notes on here. So in terms of the way that it works, or it makes its way through the body. So again, the Cercaria are going to penetrate the skin. They are going to travel through the circulatory system to the intestinal blood vessels. So that's their way. They're going to make their way through the body. So from the circulatory system to the intestinal blood vessels, and this is where they're going to mature into adults in the intestinal blood vessels. The females deposit eggs in the small hepatic portal veins of the liver. So, portal vein. The eggs are moved progressively towards the lumen of the intestine. So in the case of Schistosoma mancini, the eggs will end up in the intestine or of the bladder and the ureters, so the urinary tract of other species. And then those eggs are going to be excreted as feces or urine. So they're going to be excreted as feces or urine. Again, it depends on the species of Schistosoma as to whether this causes problems in the intestinal tract or in the urinary tract. You might recall if you watched the Parasites Eating Us Alive video, one of the things that they talked about was that Napoleon and his troops, when they entered into Egypt, that they got what was referred to as Pharaoh's curse. And Pharaoh's curse was male menstruation. Basically, they were bleeding when they were urinating. And so that was what they called male menstruation. And so they said that, when Napoleon entered into Egypt, that this prophecy came true and they had this male menstruation. But what it really was that the troops became infected and got schistosomiasis. And that caused that blood in the urine, which made it appear like it was male menstruation. Without treatment, schistosomiasis can persist for years. Chronic infection leads to an increased risk of bladder cancer because when the eggs are in the urinary tract, it causes an inflammation, and that chronic inflammation can lead to an increased risk of bladder cancer. Where this is found most prevalently, it's found in South America, it's found in Africa, it's found in the Caribbean islands, the Middle East, but not in the US. And that's because we don't get these types of snails, which are the intermediate host. You have to have the intermediate host in order for humans to become infected. In terms of diagnosis, let's put that on here. Diagnosis is made by identifying the eggs in stool or urine samples under the microscope, right? Because again, that's how the eggs are going to be released. Rarely, eggs are found in the brain or the spinal cord and can cause seizures, paralysis, or spinal cord inflammation. The symptoms of schistosomiasis are not actually caused by the worms themselves, but are actually as a result of the body's reaction to the eggs. So it's the eggs that cause all of the symptoms that you see with schistosomiasis. It's not the adult worms, it's actually the eggs. They initiate an inflammatory response. And so that is where those symptoms come from. So here are some images of these various specimens. So this is the male. You can see this gynecophoric groove, this little pocket. Notice it looks like a little fold. And that pocket is where the female would reside and they would reproduce. So this is the male. The female, again, is going to be the thinner of the two. So this is our female. Does not have the gynecophoric groove. Here is the cercaria. Again, this is the larval stage. You're looking at it under 400x magnification. You can see its forked tail. Again, looks like a mermaid tail. That part is going to break apart as the head penetrates the skin. And then we have the eggs under various magnifications. But again, you can see this little fin. That's what tells you schistosoma eggs. And so that's what you're looking for when you're trying to identify those schistosoma eggs, is look for that surfboard fin looking structure. And our last worm is going to be species of Taenia. So we have Taenia saginata, which is the beef tapeworm, or Taenia solium, which is the pork tapeworm. These are going to be a cestode, so they are a flat, appears to be segmented worm. The disease that they cause, taniasis, that is when you become infected with the adult tapeworm in the intestine. We are also going to talk about later that Taenia solium also causes a disease that's called cysticercosis, where you actually get the larval stage of the worm in the brain, eyes, and muscles. So it's a slightly different disease. It has a different mode of transmission, which we'll talk about later. And so these are the diseases caused by Taenia solium. The number of new cases in the US each year is probably less than 1,000, so it's not particularly common in the US, but an exact number is not known because the symptoms might not appear. It's very difficult to track. In terms of this worm is the only one that we are talking about that is monoecious, meaning that the male and female reproductive parts are in the same organism, meaning these worms are hermaphroditic. They have both male and female reproductive parts in the same worm. So this worm is monoecious. In terms of stages to know on the exam, so you need to know, let's put this on here, the scolex. The scolex is the head, and it has these little hooklets on it. And these hooklets are unique to Taenia solium. So if you see those hooklets, you're looking at Taenia solium. So that is the head or the scolex. You need to know that. So again, if you see the hooklets, it's Taenia solium. You need to know the gravid proglottids. These are those repeating subunits that are found in the body of the worm. And if we look at these gravid proglottids, you can see these little branch points there. Those are referred to as lateral branches. Now, I will not make you know this, but just so you know, one of the ways that we can distinguish between saginata and solium is by the number of lateral branches. Taenia saginata, the proglottids have 15 to 20 lateral branches, whereas Taenia solium has between 7 and 13. And so if we were to count the number of lateral branches, that can give us an idea of which species we are looking at. We need to know the egg, and the egg has striations. And what that basically means is if you're looking at the edge of the egg, it has like these lines looking through the edge of the egg. Those are the striations. So that's how you can identify this egg. And then we also need to identify the cysticerci, which is the larval stage that forms in the muscle. I will show you examples of our lab pictures so you can see what that actually looks like under the microscope. But there are 4 stages that you need to know for Taenia solium. Now, in terms of infectious stage and mode of transmittance. So humans are the definitive host. So humans are where the sexual part of the life cycle is going to occur. The intermediate host, where the asexual part of the life cycle is going to happen in cattle or cows for Taenia saginata, or pigs in the case of Taenia solium. There are fish tapeworms as well. There are other types as well, but we're going to focus on Taenia saginata and Taenia solium. Again, saginata is beef, so in cattle. Solium is in pigs. Now, Humans, right? Humans are going to be the definitive host. That's where the sexual part of the life cycle occurs. The adults are found in the intestine. So the adults are going to be found in the intestine. And so what's going to happen is that in feces, we're going to get eggs or gravid proglottids being released. So when somebody defecates, they're going to release eggs or the gravid proglottids, which contain the eggs. Those are going to be released in the feces. Now, when the eggs come out, the eggs can survive for days to months in the environment. So they can survive in the soil or on the grass for months. Now, the way that cattle and pigs become infected is by ingesting vegetation contaminated with the eggs or gravid proglottids. So they are grazing and they ingest the eggs or the gravid proglottids, which contain the eggs. So they're grazing, they ingest the eggs. Now, in the animal, the oncospheres are going to hatch. So here we have the oncospheres are going to hatch. They're going to penetrate the intestinal wall, right? Because they're going to be in the intestine when the animal swallows it. So they're going to hatch, they're going to penetrate the intestinal wall, and they're going to circulate to the musculature. And then the oncospheres will develop into cysticerci in the muscle. This is the larval stage. So the cysticerci is the larval stage. So a cysticercus can survive for several years in the animal. Now, the way that humans become infected is through ingestion of raw or undercooked meat containing the infective cysticerci. Because again, this is going to be found in the muscle of the animal. So in the pig or in the cattle, in the muscle, which is the meat. If that meat is not cooked thoroughly enough, right? If the meat is not cooked, well, then you're going to ingest the cysticerci. And so when you ingest the cysticerci, they're going to make their way into the intestinal tract, which then they will develop into the adults in the intestine. Now, I'm going to leave this slide for a minute and I will come back to it. But I want to show you what happens for the other disease, which is the cysticercosis, because cysticercosis is going to be different than what happens in taniasis. In terms of transmission for cysticercosis, this, remember, is the other disease that humans can get as a result of Taenia. And again, cysticercosis is going to be for Solium specifically. And so for our Taenia Solium, if you happen to ingest the eggs of Taenia Solium, you get cysticercosis. And this affects the brain, the eyes, and the muscle. And so what happens in this is that normally when you eat the larval stage in the meat of the animal, you're ingesting the larval stage, which goes to the next stage of the life cycle, which is the adult. However, if you ingest the egg, right, the next part of the life cycle is going to be the larval stage, the cysticerci. So if you ingest the egg in a human, that egg is going to hatch and you will end up with the intestine in the brain, eyes, and muscles. And so the symptoms for cysticercosis are going to be seizures, muscle or eye damage. In many cases, they might actually mimic the similar type symptoms that you would see if the patient had a brain tumor. And so what you're looking at in the bottom, these are some brain scans. And these little pockets here, these little black holes, those are all little pockets where the cysticerci the larval stage of the worm is developing in the brain. And so you can see there's a lot of them in the one on the left. You can see these pockets on the one, or I'm sorry, the one on the right and the one on the left. You can see these little pockets. Those are where the cysticerci are developing in that patient's brain. And so this can cause some severe damage to the brain. And so I have a video to show you talking a little bit more about cysticercosis.
Oleann Fox, at this time of year, we hear how important hand washing is to prevent the spread of the cold and flu. Tonight, you'll learn how hand washing could prevent a scary brain condition. A Valley mother of 4 is told that she has a brain tumor, but when doctors go in to remove it, they find something else, something living inside her brain. Rosemary Alvarez savors every minute with her kids.
That was a good throw.
Playing ball and laughing. For this mom, each day with them is a gift after coming too close to death last August.
I thought I had the flu. I started feeling tired.
But within days, this soccer coach and PTA president knew it wasn't the flu.
I started feeling numbness in my left arm, blurred vision. I'm still smiling for my kids, but now I can't get out of bed.
2 trips to the ER solved nothing.
CAT scan came back normal, no problem.
But her husband George was scared.
She couldn't eat. She could barely walk to the bathroom. So I knew at that point, she was deteriorating really quick.
It all came to a head on the first day of school as their kids aged 9 through 17 headed back to the classroom. Their mom was in an ambulance headed to Pharaoh Neurological Hospital after an MRI explained those strange symptoms.
It came back. You have a brain tumor. At that exact moment where my heart fell just completely all the way down.
But the news would get worse. Neurosurgeon Dr. Peter Nikaji.
Once we saw the MRI, we realized this is not good. It's something down in her brain stem, which is as deep into the brain as you can be.
George broke the news to the kids.
It was a rough day because it was the first day of school, and they came home with all their stuff from school and all the stuff to sign. And look what I got from my teacher, and where's mom, and what's mom? Well, mom's in the hospital, and this is what happened.
As Rosemary waited for surgery, she worried.
Oh my God, we just canceled our insurance like 3 weeks ago, and now this happens, and mostly, how are we going to pay for this?
And she prayed.
It's okay, God. If you're going to take me, but make sure my kids are good.
By the time the surgery got underway, all those PTA moms and neighbors, her kids, they were all praying too.
My girlfriend, she goes, everybody's calling and saying they're praying for you, they're praying for you. Church, they're going to church and they're having the mass, pray for you. And pray. Power of prayer. That's what I told them. Power of prayer.
It worked. Dr. Nikaji was expecting to find a brain tumor. He showed me the video as he operated on Rosemary's brain, carefully working, knowing this is the most dangerous spot in the human body to work next to, the brainstem.
Here's the lump, and at this point it still looks like a brain tumor, and so we're just opening up into the inside.
And then the unexpected. No tumor, instead a worm. Watch as Dr. Nikaji grabs the worm and gently tugs it out of Rosemary's brain. It's still alive. You don't hear him on the tape, but Nikaji actually chuckles.
Which I'm sure is a very strange response for the people in the operating room, but because I was so pleased to know that it wasn't going to be something terrible.
This worm comes from eating undercooked pork, but more frighteningly, it can be spread by people who have the parasite in their bodies and don't wash their hands well after using the bathroom. That is the problem.
And it only takes one person who is spreading it constantly to get a lot of people exposed, and some of those people are going to go on to develop this problem.
It's impossible to pinpoint exactly when Rosemary contracted this brainworm.
Probably she was one of these very unfortunate people who had somebody handle food for her, that she ingested it, and it got to her brain.
What is normally a rare occurrence has become much more common. Dr. Nikaji has removed 5 of them in the past few months, and other doctors at Pharaoh, they're seeing more of them too.
I think it could be a fluke, but it does concern me that if we see more and more cases, as one person is infected and starts to spread these, it spreads it to other people who become infected, and then they spread it. so that more people are at least at risk.
Meantime, George and Rosemary are stunned, relieved, and a bit creeped out by it all.
It could have ate holes in her brain like a apple.
This whole part of my head, from here to here, it's numb. I just learned to live with it.
Rosemary shows off the scars.
I had to learn to walk, to balance.
But Rosemary says the most important thing she learned is how to really live.
And so what you saw in that video was that the way that you get cysticercosis is different than the way that you get taniasis. And again, that's because when you eat contaminated meat, you're ingesting the larval stage. And when you ingest the larval stage, it's going to form the adult in the intestine. However, if you ingest the egg, think about where the egg is coming from. So in the video, they were talking about how if somebody is infected with taniasis, if they have the adult worms in their intestine and they go to the bathroom and they don't wash their hands well after going to the bathroom, right? They go to the bathroom, they don't wash their hands. Feces, remember, has the eggs on it. And so if they happen to have eggs on their hands and now they're preparing your food and you ingest those eggs, the next stage in the life cycle is going to be the larval stage of the cysticerci, which likes to form in the brain and muscles. And so this is why you end up with cysticercosis. So it's a different disease from taniasis, and it happens by being infected by a different stage in the life cycle. And specifically, it's ingestion of the eggs that is going to lead to cysticercosis. And this is for Taneous solium, just like the hooklets are for Taenia solium. So now we need to look at the rest of the table for Taenia solium. And so the approximate size of the worm. Now again, you do not need to memorize these numbers, but I did want to show you so that you could see how big these get. So for Taenia saginata is typically the larger of the two. Usually these worms are up to 5 meters, which is approximately 16 feet long. So they can be up to 16, or they're usually up to 16 feet long, but they can be as large as 25 meters or 82 feet. So just think about that for a minute. 82 feet is how large one of these worms can get. For Taenia solium, they're going to be smaller than saginata. They're typically between 2 to 7 meters or between 6 to 23 feet. So again, these are going to be our very large worms, right? They're much, much larger than the other worms that we've talked about. So these are very large worms. In terms of our symptoms. So for our symptoms, usually there are no symptoms or mild symptoms. So many people are asymptomatic. Symptoms typically are worse with Taenia saginata just because of its larger size. If we do have symptoms, it can cause abdominal pain, loss of appetite, weight loss, and upset stomach. The most visible symptom, or the thing that you can see that might indicate that the patient has taniasis, is the active passing of proglottids through the anus and in the feces. So if you were to look at somebody's feces who is infected, you might be able to see those gravid proglottids in the feces. In rare cases, the tapeworm segments become lodged in the appendix or the bile and pancreatic ducts, which causes more issues. And again, we also talked about the symptoms of cysticercosis, which is that, and that was on the next slide, which was that it can cause seizures and muscle or eye damage. In terms of our treatment is praziquantel, just like for schistosoma. So that's the drug of choice. Like niclosamide is an alternative drug, but it's not effective against cysticercosis. And so it's not used as much in the US. It is an ATP synthase inhibitor in worms, so it prevents ATP synthase in worms. But again, praziquantel is typically the drug of choice, especially here in the US. Now, in terms of the way that this moves through the body. So, when we ingest the cysticerci, right? When we ingest the cysticerci and it goes into the body, the cysticercus develops over 2 months into the adult tapeworm. And so the adult tapeworm is going to survive for years in the intestine. And they're going to attach to the skull, or attach to the lining of the intestine by these suckers. So they have, whoops. They have suckers these suckers on the head, and these suckers on the head allow them to attach to the intestinal wall. And so when they attach to the head, they're going to grow in the intestine. Diagnosis is made by identifying eggs or proglottids in a stool sample. So looking for the stool sample, looking for the eggs or the proglottids in the stool sample. Individuals will typically also be asked if they've passed tapeworm segments. So could they actually see the visible tapeworm in the feces as well? Stool specimens can be collected on 3 different days and examined in the lab for eggs using a microscope. Tapeworm eggs can be detected in the stool about 2 to 3 months after the tapeworm infection has been established. So it takes about 2 to 3 months from when you ingest the worms to be able to start to see those eggs. The adults, when they're in the intestine, they produce these proglottids, which mature, become gravid, and then they detach. So notice they're detaching from the tapeworm and migrate to the anus or passed as stool. Approximately 6 of those proglottids are released per day. Taenia saginata will release up to 600,000 eggs a day, and Taenia solium will produce up to 300,000 eggs per day. In terms of the length of the worms, so Taenia saginata adults usually have between 1,000 to 2,000 proglottids, while Taenia solium will have an average of about 1,000 proglottids. Now, where are these types of helminth infections prevalent? Where do we see taniasis? So beef tapeworm is most common in Eastern Europe, Russia, East Africa, and Latin America. Pork tapeworm is most common in Latin America, Eastern Europe, Sub-Saharan Africa, India, and Asia. And so this is where you would see these most often. Now, in the video for the cysticercosis, they have had outbreaks in the states that border Mexico, because in Mexico, this type of infection is more common than it would be in the US. And so the states that border Mexico are more likely to see these infections than the rest of the country as a result. So here are the images that you need to know for the exam. You need to know Taenia solium head, which is the scolex. So these are the sucking disc. And so this allows the worm to attach to the lining of the intestine. And again, you can see those hooklets, and those hooklets are going to help Taenia solium attach to the intestine as well. So you need to know the scolex, the head. You need to know the cysticercus. And so again, this is the larval stage that's going to develop in the animal. And so you can see over here, you can see Cysticercus. So that's the whole larval little worm sac that you're seeing there. And then on the right is it zoomed in. And you can see the black little hooklets already that are forming on the scolex of the Cysticercus. So that is how you can identify this as being Taenia solium, is seeing those hooklets. So this is going to be your larval stage, the Cysticercus. Then we have Taenia pisciformis, so just a different species, but the same idea. We have our gravid proglottids, and so you can see these little lateral branches. You can see the eggs within these structures. And so these are what can be passed in the feces. And again, this is what also contains the eggs, which then can become infectious. In terms of the eggs, so what you're looking for, again, you don't have to memorize that this is Taenia pisciformis. Just know that this goes with Taenia because the Taenia solium eggs look the same. But again, what you're looking for is the outer edge of this egg has those striations, those like lines that go around the egg. And so that is what you're looking for to identify the Taenia eggs.